And we're back.
Five? We have already collected five? Okay, we're on six. We're on six.
Welcome to the Octopus Odyssey Expedition on board the research vessel Falcor 2. We are a couple hundred miles, 100 miles south Costa Rica. We're approximately 150 miles off the Costa Rican port of Punta Arenas, where we are exploring a series of seamounts in the Costa Rican Pacific. We've been here for over a week now and made a couple of very interesting discoveries already. The whole point of our expedition is to understand the influence of physical, geochemical, and microbiological processes on the egg brooding behaviors of the charismatic deep sea octopus that live in this area. These octopus females tend to brood their eggs at sites where there's slightly warmer fluids emanating from cracks in the rocks, again, near the summit of these seamounts or outcrop structures. We think that's fascinating, so that's what we're out here doing. We are doing this thanks to a very international team. I can count at least 6 nations that are represented in our scientific party, perhaps more nations. We take into account the technical staff and the ROV team of engineers. And our arms. Good? Please good? Yeah. Our arms, eyes, and our presence, really our avatar at the seafloor that allows us to do all this science is a fantastic piece of equipment called SuBastian. So this is a remotely operated submarine vehicle whose arms you're seeing at work there. This SuBastian comes along with all of its friends, a couple dozen engineers and ROV pilots that keep operations running smoothly, generally on a 24-hour basis. So we've, on the suggestion of our octocoral expert, Odeliska Breedy from the University of Costa Rica, we've just collected quite a complex specimen of a beautiful pink octocoral hosting a couple of stars there. Seems like this might be too complex of a shape to fit into our quiver. Do we have a bio box available? You can't use a bio box?
I don't know. You should ask him. Can I ask him? Yeah. Do something to check it out. No, it's not possible.
So for everyone in the audience, we're trying to figure out the best possible way to collect this this sample. Let me ask. Do we have any rocks in bio box?
Rock on the fireballs up here.
Maybe we can use those two. Then we have these 2 spaces.
Can I ask Beth? Should I?
Yeah, we should ask Beth, but I know that's open and that's open, so potentially good language centers. We got a confirmation from Beth.
It might go in.
Might go in? Okay, all right.
Let's try it for the big sticker.
Just given the time, you know, like.
Wow. Nice.
I don't think we're gonna.
That's me trying to put a umbrella back in its case.
Oh, it's happening?
Love it. Just put the stop on it. I mean, it's not going in the aquarium.
Got a question for the coral expert here. Oh, got someone in the chat asking about.
Is this calypso for which medium this is? No, I don't think that it's calypso,
Okay.
but I have no idea what this is.
Okay. Just to answer Connor here, we don't think this is Calyptophora, but Yeah, I'll ask the expert in just a second. There's,
When take out this.
yeah. We'll request more of Oda's time in a second. She's in the middle of a collection, and I wouldn't want to distract from the action.
Many people can identify from seeing this, but it's important to take it to the lab and analyze the splice and the structures.
Okay. Do you want to tell me that?
Oh, you can tell them.
Yeah. Okay. We got the clear from Beth. All spaces are available in any of the buy boxes. Yeah.
Okay, just in case.
For future collections, yeah.
For identification of the octocorals, it's better
Sorry,
to
Oda, can you identify yourself?
take them out. What?
Can you identify yourself?
Yes, I'm Odalis Cabridi. I work with octocorals, especially shallow water. Now I'm exploring the deep water octocorals in Costa Rica with these explorations. And I prefer to take them out and study the sclerites, that are the structures that make the skeletons, in order to give a accurate identification of the organisms. Thank you.
So again, that was some very welcome commentary from our octocoral expert, Professor Odalis Cabridi from the University of Costa Rica. Yeah, we appreciate that, Oda. Thank you. This is the most exciting part of the dive for her, and she's very hard at work identifying the next set of specimens for collection here in the few minutes we've got left.
All right, looking around.
You see that at the bottom there is a small thing. Can you?
This here?
This side. Here.
Oh, okay.
This
See him there.
Here?
yes. It is easy to collect that. Nothing.
You are welcome, Connor.
We already collected one of those, but I would like to have another one because I think it's a new species. So you can collect this one.
And thank you for being such an avid fan of our dives here. Yeah.
That one is in the genus Swift,
Okay, which one? The red one?
the red one. And I know that because I'm working with that genus before. You can tell that. You can see.
So that small octocoral, the small red octocorals in the genus Swiftia, according to our octocoral expert here. And yeah, she's definitely familiar with that one. I guess a lot of her publications are based on that one. So very confidently identified the genus just visually.
Want to get the quiver open first and then we'll look forward?
We'll take some time here to give a general summary of the action over the past half hour or so in Spanish language commentary. Buenas tardes y bienvenidos a todos. Me llamo Gustavo Ramírez, soy un profesor en el Cal State University en la ciudad de Los Ángeles, en California. Y bueno, me encuentro a bordo del buque de investigación Falkor 2, que es un buque operado por la organización Schmidt Ocean Institute, y estamos en el Pacífico costarricense estudiando montañas submarinas donde hemos encontrado colonias de pulpos, de hembras pulpos que básicamente ocupan estas cimas de los montes submarinos para dejar sus huevecillos y para cuidar sus huevecillos. O sea que es algo súper interesante. Parece que hay una asociación entre el lugar donde las hembras escogen para dejar a sus huevos. Usualmente dejan como 6 o 10 huevos a la vez. Hay una asociación entre sus preferencias para dejar los huevecillos y el fenómeno de agua hidrotérmica que sale de las piedras. O sea que usualmente donde hay pulpos hay cantidades muy grandes de agua hidrotérmica a una temperatura de más o menos 10 grados centígrados más alta que el resto del agua del mar. Y eso es el fenómeno que venimos a estudiar aquí. Estoy acompañado por un grupo fantástico de expertos en biología marina, expertos en geoquímica, expertos en la física del océano y bueno, todos estamos aquí para tratar de describir este proceso muy impresionante que dicta la ecología de estas montañas submarinas. Bueno, empezamos nuestro trabajo hace más de una semana ya en una montaña submarina llamada El Dorado, que ya se había visitado justo hace una década más o menos, una década o 10 años, una década o 9 años, se había visitado El Dorado. Y ahí es donde se encontraba la mayoría de, o se encuentran la mayoría de las hembras con huevecillos que habíamos que acabo de escribir. Esas, y bueno, nosotros pudimos confirmar que siguen ahí. Wow, yeah. La fuerza del coral ahí, que bueno, al tratar de sacar el ejemplar del coral, hemos traído una piedra también. Muy fuertes estos, este cemento a la base del coral. Pero nos estaba diciendo que en El Dorado pudimos ver a las hembras y confirmar que seguían ahí y que la comunidad seguía muy feliz porque vimos docenas y docenas de pulpos que acababan de nacer, de bebés. Bueno, sabemos que hay muchas otras montañas submarinas alrededor de esta pequeña montaña o outcrop llamada el Dorado. Y bueno, parte de esta expedición son 17 días dedicados simplemente a la exploración de las otras montañas submarinas alrededor del Dorado. Estas montañas se saben porque tenemos mapas de sonar medios un poco anticuados, o sea que sabemos que hay cosas sabemos que hay montañas en el lecho marino, pero nunca las habíamos visto a este nivel de resolución, con esta intimidad, ¿no? Ahora hemos explorado 3 lugares en los últimos 4 días. Oh, ok, we got the rock. Yeah. En los últimos 4 días, 3 otras montañas que nunca se habían explorado por los humanos antes. Una de esas montañas ni siquiera tiene nombre. O sea que hay varias conversaciones que se han tenido con los científicos líderes aquí de la expedición para empezar a pensar qué es lo que queremos llamar esas montañas sin nombres. Nuestros colegas costarricenses ya tienen varias ideas ahí que representarían algo de la cultura de Costa Rica, ya que estamos en aguas territoriales de Costa Rica. Es importantísimo esta expedición para los costarricenses, ya que 90% de su país se encuentra en el lecho marino profundo. Y bueno, después de estudiar esa primera montaña, por primera vez en toda la historia de la humanidad, fuimos a otro lugar, a una segunda montaña que nunca se había estudiado y bueno, ahora estamos aquí, 5 días después, en una montaña que se llama Tengosed, que también nunca se ha visto con ojos humanos. Esto es nuestra segundo buceo en Tengosed. Estuvimos aquí ayer y estamos aquí hoy. Y bueno, la montaña se llama Tengosed porque es una estructura gigantesca de 6 kilómetros de diámetro que domina el paisaje aquí verdaderamente y por ser un poco más fría, de acuerdo a los datos de temperatura que tenemos del área, es hipotizado, la hipótesis es de que esta es una montaña donde la densidad del agua sube y por ser más densa el agua baja y se penetra dentro de estas rocas y entra en la primera capa de la litosfera. Eso es más o menos la física y oceanografía que nos interesa describir mejor y es una de las motivaciones para este crucero. Bueno, regresaré a mis descripciones en inglés. Si hay algún hispanoparlante en la audiencia, por favor, mándenos saludos de dónde nos están sintonizando. Y cualquier pregunta, estoy aquí para responderla, junto con otros especialistas. O sea que puede poner en el chat inglés, español o incluso portugués, ya que tenemos y francés, sí. Con suerte y alemán, ¿no? Bajamos al chef también. Sí, bajamos al chef, sí. Anyway, so we just witnessed a collection of just wonderful, small but definitely wonderful red coral here. And we were all surprised to see that when we grabbed it at the base, rather than detaching, it brought an entire rock along with it. So that's quite remarkable and unexpected. Yeah. Ok, saludos a Gonzalo Jiménez, que nos sintoniza desde Kiel, Alemania, el norte de Alemania. Sí.
Let's go see that anemone, huh? The crowd.
Now we continue our survey here at the summit of Tengosed Seamount. See a complex geological view here with a lot of black rocks, which I think are basalts, that serve as attachment points for a lot of these echinoderms, right?
Okay.
Or corals, right? Yeah, great diversity of corals. We see a sea star in the back, a couple of sponges. Do we want to collect the sponge?
Yes. But
Yeah.
not this
We
one.
want the ones that hang, right?
Yes.
Like the ones that look like a Pokemon.
Yeah, you see,
Yeah. They're definitely around here, though. They're close somewhere.
what time would I have to go?
I think our time's up at 4 o'clock. Because is that right, Scott? We're gonna need about an hour and a half, right, to bring it back up.
I'll have to confirm with Carl.
Okay.
So what time are we supposed to be off
So
on?
this is the nursery.
Yeah.
3-ish. Adam should be here any minute. I can let him confirm that.
Oh, okay, yeah. Sorry, I have some LA time.
You're on LA time.
All right. Oh, this looks like we're getting close to the summit. This looks like where we were yesterday, and we're on the lookout
Can
for.
you look to that coral in the left? Here.
This one right here, Olga?
Yes. See.
You can see
Is that
all
what we're
these
talking about?
skate eggs in the foreground here.
Yeah. Oh, plenty of skate eggs here.
Yeah.
Oh, it's
These
full
look a
of-
lot healthier too. You know.
This is much more like I expect from a nursery. Yeah.
This is what we saw yesterday, right?
Not quite this dense.
Even more dense here.
Oh yeah, this is much denser.
Okay. Wow. This one right here. Yeah.
A bubble gum for it?
Wow.
Yeah.
Looks like it's Aragorn. Bubble jump coral.
Is that what they're called?
Bubble jump coral.
Bubble gum?
Sí,
Really?
de chico.
Wow.
Considering how unsedimented these rocks are,
Mm-hmm.
and that there's all these skate eggs,
Yeah.
it would be good to pull out the high T probe
Yep.
and just kind of poke around and see if it's a little bit warmer. Because on the recent expedition out in Canada, they found an association of skate eggs with venting.
Fair enough.
So
Yeah.
this is a,
Yeah.
I hope you're getting lots of pictures, Myla.
Yeah.
All right, so I'll just land here and.
So this is an extremely exciting site.
We'll poke it.
As I previously mentioned, there appears to be an association with octopus egg brooding behavior and hydrothermal venting at these seamounts. This is information that I'm just getting from our chief scientist, Vatorka, here. A similar association between egg brooding, or I guess egg laying, and the hydrothermal venting has also been observed for skates in the northeastern Pacific around Canadian waters. So there's a good chance that this skate nursery at the very summit of Tengosed Mountains can be a marker for potential hydrothermal vents exiting at this site. Again, we're dealing with low temperature fluid flux here, so that means that it's not as dramatic of a water exit there as it is with black smokers. You know, black smokers look very cool. They're the picturesque sites for hydrothermal venting, but they're not very representative. Most of the subsea floor circulation and venting that happens worldwide happens in very, very low energy or low temperature fluid flux. So that's exactly what we're looking for here. Again, not as dramatic of an event, but a much more universal one. You'll see our pilots manipulating that camera to basically focus on small crevices where venting may be happening. And there's indications of or we're hypothesizing that perhaps skates are using this site where venting could be happening to lay their eggs.
Hello.
Show us the minute.
There's no venting here. Go away.
So this is again a very intimate look into the nooks and crevices of the summit of the seamount. This would be impossible to do with the more standard oceanographic techniques for the detection of hydrothermal activity, which is basically lowering giant thermometers from a ship and stabbing the seafloor and measuring the temperature there. Again, we're fortunate enough to be at the seafloor and being able to zoom in on orifices that could be as small as just a few centimeters square in diameter there, and just waiting to see if we see the shimmering. We know that shimmering can be observed when the water exiting is at least 1 centimeter, 1 degree Celsius warmer than the background, which we've seen on this mountain, on the seamount earlier today. That's what we're looking for here. It's pretty one of the most exciting parts of the dive here.
I think we should just go ahead and start measuring. And then if we see a response, then we'll zoom in on it, just because we don't have a lot of time.
Okay.
Do not think we should sample one of these. Do not. We picked one up yesterday as empty.
Okay.
We already got one.
Yeah. Just leave that.
What's the temperature right here? 3.22.
Down at the base.
Yeah. Yeah. Well, Beth wants a temperature reading. Yeah. So after that, it's up to their discretion, but there's nothing else but corals here.
I can foretell a lot more.
So the next order of business here is going to be to bring out a device that allows us to take a temperature measurement. This, that's what you see the ROV pilots masterfully manipulating this here. And, again, we may not be able to see any shimmering, but it does not mean that there isn't water, that's slightly warmer than the rest of the background ocean emanating from here. And again, this is kind of what we're hypothesizing based on the prevalence of skate eggs that are clearly visible at this part of the summit of Tengosed Seamount. So this probe has I guess a thermometer at the end that allows us to take a temperature, and we're going to insert it into one of these cracks in the rocks or small orifice where there could be water exiting here. And again, we'll know if water is exiting just by knowing the temperature, because warmer water will be less dense than the surrounding colder water. So if we get a temperature reading, that means that there is potentially hydrothermal venting at this site, low temperature hydrothermal venting.
I think so. Yeah, we may have to afford. Okay, no problem.
So it seems that the background seawater is currently at a temperature of 2.36 degrees Celsius. And we are curious to see if our reading from the probe that we're going to insert into one of these crevices in the rocks is at that temperature or perhaps slightly higher. If the temperature is slightly higher, then again, that would be an indication that there's potentially warm fluids exiting at the summit of this seamount, Tengosed Seamount, and that's the operation that's going on now.
temperature readings. No visual.
And would it be on deck at 5, still thinking 3, or a little bit later?
Well, I mean, it's like around the top. Okay.
But I just saw there are a couple of eggs just beneath the box that I think are still with the rain inside
Okay.
because of the color. I was
Okay.
reading the paper from this week.
Yeah. Okay.
We see our ROV pilots here skillfully inserting this probe at the seafloor, 2,000 meters beneath the waves where we find ourselves, and taking a temperature reading. So far, there's some variation by about 1/10 of a degree above and below the background temperature. That's 2.36 degrees Celsius. The larger number they see overlaid on the screen there shows the temperature that's being recorded by the probe that was just inserted into that orifice there. Again, at the summit of Tengosed Seamount. We're gonna go ahead and poke around here, maybe try another site. Hello, Mr. Crab.
I think these are carnivorous.
So again, our probe is measuring more or less the same temperature as the background seawater so far.
Nope.
Look, and see, this looks like the one you see in.
A little shrimp.
Do you think it looks like the little yes?
Yeah. Albinocaris.
I think I did that. Yeah, that's great. Yeah, that's my turn.
Look, there's some more.
We're trying one more place here with a temperature probe.
So they're 2 times.
One more orifice here to see if there's any venting.
White ones and pinky red ones.
Again, the larger overlay they see on your screen is the temperature that's measured at the tip of this probe. And it is not, as far as we can tell, significantly rising or significantly higher than the background temperature they see in the smaller left-hand column there at the bottom where it says temperature. That's 2.36.
All right, as you can see, we haven't gone anywhere just yet. We scooted over a smidge and we're checking out some more of these holes in the rocks for any temperature variance.
We back on, Alex?
Mm-hmm.
Okay.
Yep.
Welcome to everyone tuning in from home. It's either through YouTube or Facebook or whatever other social media channels. My name is Gustavo Ramirez. I am a professor of microbial ecology and computational biology at the California State University in Los Angeles. And I've been fortunate, quite fortunate to find myself in the middle of the Pacific Ocean, about 140 miles off of the coast of Costa Rica, to be more precise, as part of a scientific expedition that's in exploring seamounts. So these are mountains at the seafloor.
Can we pick up and maybe like look around and like see
Yeah. Today we find ourselves in a pretty large seamount by relative standards. Really a giant of the seafloor, approximately 6-kilometer base called Tengosed. That translates to "I am thirsty" in Spanish. And Tengosed is hypothesized to be a site of hydrothermal recharge. So this is a relatively colder rock that comprises this big seafloor edifice. And this cold rock is hypothesized to predominantly uptake water or drink water, hence the name Tengosed, from the background ocean, and incorporate that water into warmer crevices under the seafloor, with more lithosphere buried beneath the sediment. Then that water flows through the subseafloor crystallograpers and eventually exits at sites where there's an opening and the water is warmer, meaning less dense. This process is called hydrothermal circulation, and it's extremely important for setting the physical, chemical, and biological equilibrium of the ocean. The entire volume of the ocean, as we know it, cycles itself through these subseafloor crustal environments with a periodicity of once every 300,000 years, which is the blink of an eye in geological timescales. So this process determines the chemistry of the ocean. Of course, the ocean interacts with the atmosphere. So there's ultimately, this process may also influence the chemistry of the atmosphere and the overall oxidation reduction state of our planet, which is something that is extremely important for dictating the arc of evolution over millions and perhaps hundreds of millions and billions of years on our little planet here, planet Earth, our wet, rocky spaceship. Oh, was this our big anemone from yesterday? Is this another one?
No, because I think he was right at the. I think
Perhaps, right?
it might be actually.
Yeah, yeah.
Rock, wasn't he?
Yeah.
Oh, maybe it's this one over here.
Yeah, it was
Whoa, man, that's so cool. You got a couple frame grabs? This is just. Okay. Wow, 12,000 days. Wow. So was that like 4 years, 5 years? Okay. One of my colleagues here just brought up an interesting fact that I should share with the audience. We suspect that there is a sea skate nursery here, given the disproportionately high number of sea skate eggs that we're seeing at the summit of Tengosed Seamount. The fact that was just brought up is that these deep sea skates have some of the longest egg incubation times in, I guess, cartilaginous animals, right? Or cartilaginous fish. Yeah. So they can their eggs may take up to 12,090 days to hatch, which I guess the rough calculation here is between 4 and 5 years. So it's quite incredible what we're seeing here. We are navigating towards the actual acme of this Tengosed summit at this point. We were here yesterday just briefly before wrapping up yesterday's dive. And again, what you're seeing here is just surreal. We're about 1,000 meters above the seafloor here at a mountain that's completely submerged. Um, and at the very top of this mountain, when we were exploring it yesterday, we found this giant, perhaps meter or meter and a half diameter sea anemone that just resembled a crown, really, at the very top of the seamount. And it's like this animal just proclaimed itself king of the hill. Um, oh, it keeps rising. It keeps rising.
Yeah, it's more still on the ground,
Okay.
but it's too sedimented for them to.
Oh, yeah. Big guy.
There appears to be an uptick in the, I guess, technical terms would be alpha diversity that we're seeing here. Many different kinds of species present in this in the vicinity of the very summit of Tengosed Seamount. See this gorgeous sea fans? We've been seeing them a lot throughout this dive. Some sort of what is that? A crab? Numerous sponges. And again, paradoxically, both an alien vista here and something that's very familiar too. The colors really pick up here. It's quite a beautiful contrast that we're seeing here. Again, prior to yesterday, no human had laid eyes on this gorgeous seafloor structure.
But first I want to zoom. Just to get it. There's no shimmer.
Everyone's eyes are carefully watching this screen as we zoom into crevices at the very seamount here, looking for shimmering water, which would be an indication of a hydrothermal discharge at the summit of Tengosed Seamount. Wow.
Is the current pushing it at all?
It's moving, but it's not really. There must be some current.
Yeah, it seems like there's coming some
Mhm.
west.
Yeah.
Again, just another breathtaking view of the summit here. See a couple of sea stars now. With the larger fragments of jagged rock now. And different coral types. And we are on the search for shimmering.
got these little worms that are usually good indicators. Can you pause here for a second here? Got all the right indicators here. You got the little worms, the shrimp.
We've got the probe on there. We just
Yeah, we got the probe accessible. Let's try to get under there and see if there's anything.
Did we figure out what the tide was?
It's not low tide.
Is that
It's past
low?
low tide.
what's the variation type here? Like less?
I don't know. But it's enough that when we put down pressure sensors and temperature sensors before and oxygen sensors, we could see a daily cycle in the flux of temperature and oxygen coming out of the sites.
Which one was that?
Un saludo muy especial para todos los amigos y familiares que nos sintonizan desde mi ciudad natal, igual a Guerrero, México. No solo quiero recordar a la audiencia que cualquier pregunta que pueda haber acerca de nuestras actividades aquí en el Tengosed, en la montaña Tengosed, por favor, escríbanla en el chat. Y bueno, si es la mitad de la gente que está aquí es hispanoparlante, o sea que fácilmente, si no la puedo responder yo, puedo encontrar algún experto que pueda clarificar cualquier tema.
I know. It's got all the right looks. It just doesn't have any temperature. Unless like the whole thing is leaking and we just can't detect it because we're sitting on it.
You thinking like the ambient, the water be like 1.8 and this is 2.2?
Well,
It's
we're,
2.1 up to 2.49.
yeah, it's been doing that
Yeah,
the whole
it's
time.
yeah,
That's kind of its range.
States now. States now.
2.3.
Okay, still no anomaly. Okay.
Let me have a look around this area though. This is what you're looking
We continue the search for slightly warmer water temperatures in the background seawater. This particular site that we were just at appeared to have all the indications of a potential hydrothermal fluid venting. There's a couple of shrimp that like to hang out there, some worms, and where we've seen those animal markers, we've seen venting oftentimes. So that's why we took that temperature measurement. But we've got a couple of minutes left on the dive here, and we'll use this precious time at the summit here to microscopically basically search small crevices where we may find indications of slightly warmer fluids exiting from the mountain here, from the seamount.
Are those worms on that one?
Yeah, and it's the same indicators. It's
It's
just,
just older, but that one's got sediment like you said, right?
yeah. And like when we saw that 1 degree anomaly in the other place, like it was
Wow.
clearly shimmering.
Yeah.
Yeah.
Yeah, you see it.
There's the King of the Hill.
That is the king of the hill. Can we get a speed run, please? Yeah. Wow.
Try and throw. It's just the sediment though. Sediment. That's what I'm saying.
There might be some venting there.
back over all the skate eggs.
Yeah. This is exactly where we were before. So
Mm-hmm.
I recognize that coral.
Yeah.
And that pile of skate eggs.
Yeah. I mean, there's a lot. Like,
Yeah.
the skates know something.
That's your secrets.
Did you do some Niskins here?
No.
Should we do Niskins? Sure.
Gorgeous bubblegum coral there.
How intermittent?
What's that? Oh, the probe. Should I? I'm gonna go back over that rock.
Couple minutes we got left of this dive.
I mean,
We're
this is
gonna
where the skate eggs are, so it's
we're
probably-
gonna collect some seawater using something called Niskin bottles. These are cylinders that can of 2 and 5 liter volumes, I believe, that have open ends and are attached to the top left end of the ROV. That open cylinder can be closed at any point during the dive, and theoretically, well, I mean, or factually, the closing of the top and the bottom of the cylinders brings water from the depth where the sampling was made, right up to the surface, where we can then sample it once it's back on deck here. So what you see is the manipulator arm stowing away its that temperature probe and basically freeing itself to then go over to the triggers for the Niskin bottles.
Is it like a food source for the skates or something?
They eat?
I don't think there's anything in the water that's different.
I mean, they have a yolk in there with them. They have a yolk.
Yeah.
But if you're talking like a hatch time of 4 years, if you can speed that up with warm water, then
It's 4 years.
it can be for the deep water skates, yeah.
Wow.
12,090 days, according to Wikipedia.
Wow.
Yeah.
Hey, lots of time to prepare.
12,000 or 1,200?
1,200. I'm sure. Yeah, 1,200. Thank you.
1,000 is a.
That's more than 4 years. Yeah.
Which Niskins would you like?
Number 3 or one, 2 or 3?
What's the volume on the Niskins? 2 liters and 5 liters?
Yeah, 2 is,
That was a 5 liter.
Okay.
and then the 2, 5 and 6 are the 2 liters.
Can you do also 6, please? 5 liters.
Number 3. Okay, so which one was the 3? 3 was a 5 liter. Yes. And then second.
Okay, so what we saw there was the ROV arm pushing the triggers to close 2 Niskin bottles. Again, those Niskin bottles are cylinders that are open. When we push the trigger, the top and bottom of those cylinders are closed, and they bring with them approximately 5 liters of water collected at this depth all the way up through the rest of the water column and back up to our ship, where we're then able to analyze it. There's many different people on the ship that may have different analysis that they want to do in these water samples. Microbiologists may want to filter microbes that live in the deep ocean here onto a filter that then they can use to extract nucleic acids and then analyze based on DNA, analyze the microbial communities that live here. Geochemists may want to look at the major and minor ion compositions of the water at depth versus the water at the surface. So everything that we collect here is definitely used and welcome and satisfies our ultimate goal of a better scientific and ecological understanding of seamounts in this area. Why is that extra white? Yeah. That's
the light
cool.
colored one.
Yeah, I think
Yeah.
the one you're.
What do you see?
Yeah, well, those are hatched already.
Yeah.
See that light colored one? You see the red ones? So can we put this in a bell box?
Which one are you going for?
Little one there?
So I think that one has sediment on it.
Mhm.
That one's broken.
Yeah.
So,
Even though it's narrow, but
yeah, this isn't a good,
Okay.
these aren't good. Let's keep looking.
You looking for the green ones?
for the brown ones, like light brown.
Next to the crinoid.
I don't know what that was. The bell box, okay.
Either
This one.
quiver or biobox.
This one.
Oh, yeah.
Here's one.
Yeah, there's one, that one.
That one looks good too.
It's a good shiny one.
It went really well in a coral quiver, the one we looked at earlier.
Yeah, yeah. I was just saying that there's already one, maybe we could put the same one, or?
I think we want to keep this.
So they're currently looking for one more skate egg to grab here. And now that we've found where the skates clearly seem to prefer laying their eggs, I think the idea is that these might be a bit healthier or at least different, since all the skates are laying right here versus the one we collected earlier, which was kind of before we actually got into the mass of them.
Oh,
Every dive leading up to this one. So again, we've got some pretty cool looking rocks with what might be like really dark spots that could be manganese oxides, and or red spots that could be iron oxides on the surfaces. Very weathered, and our goal is to analyze them. But again, results are still TBD. We're gonna have to extract the DNA and do some high throughput sequencing. But yeah.
Mila, can you get some highlights, please?
trying to expose the corner that I grabbed. Do you want to do the light trick?
No, I think we can just go and put it in. We're either taking it or we're not. So I can go in Biobox 1.
It's gonna be biobox?
Nice coloration on it.
Yep.
Can you tell age by looking at it, or you don't?
You know, they look like ravioli. It's one of the comments in our chat. Ravioli.
It's almost 3:30. This one probably need to come up.
Alright. Fly over the top there.
Coming up in the last minutes of our second dive to Tengosed Seamount. Got plenty of samples collected. Again, we'll use the last couple of minutes here to just do another flyby of this very interesting environment. That's really extremely diverse in terms of life and geology. Our last flyby here before departing for the surface world 2,000 meters above.
When you get to the edge of this, just depth. I want to see if the water goes down.
Are you watching the
Yeah.
Cool.
I'm not watching that one, but I'm watching this one.
Let me know when you would count off bottom.
Sure.
And then, are you good for viewing of cameras on it?
Go ahead. Playa espinosísima. Micaela, no estamos 100% seguros de qué especie de skate estamos viendo, pero mis colaboradores tienen sus sospechas de que es batirraya espinosísima. Sí.
Yeah, I'm not seeing any appreciable difference. She's not paying attention. Yeah. All right. Thanks for letting us get those final samples.
On our way up. Okay.
But I
All right.
think-
Our, all right, so this concludes our second dive, both dives culminating at the summit of Tengosed Seamount. Again, this seamount is a behemoth by relative standards. It's about 6 kilometers in diameter, and it rises to about 1,000 meters above the rest of the seafloor. We are the first explorers to really lay eyes on this structure that we believe plays an important role locally by being a site of hydrothermal water recharge, meaning drinks up water from the surrounding and incorporates it into the crust. And then as the water moves through the crust, it evolves and emanates in other places when it becomes warm. So long story short, my name is Gustavo Ramirez. I'm a assistant professor in microbial ecology and computational biology at the California State University in Los Angeles. And on behalf of the SOI ROV team and the rest of my international collaborators on board the Octopus Odyssey Expedition, we are officially signing off. Thank you all for joining, and we look forward to seeing you at our next dive that begins at approximately 7 a.m. Costa Rica time tomorrow. Place to be explored, to be determined, but what's not to be determined is what's guaranteed is we'll learn something that hasn't been known about this extremely intriguing environment. So again, thank you all. Take care.
And that brings us to the end of the dive, as that wonderful sign off just said. But we've got some time to the surface, and I think we'll do another blue water ascent today. So mics will be off, but if you want to stick around, you want to watch as we go back up through the water column, more than welcome, and we appreciate y'all exploring with us. All right, folks. This is Alex just jumping back on. Thank you all so much for exploring with us, and those of you that stuck around and rode to the surface. Got a couple nice things in there. We are just about 100 meters down. We've got our floats on the surface, and they're going to begin recovery. And with that, I'll sign off for this particular dive, and we're hoping to be back in the water again for the same times tomorrow. So enjoy the rest of your day, evening, or morning, wherever you might be, and thank you for exploring with us.